All right, I'd like to introduce today's speaker, Arjuna Rajakumar. He comes to us today virtually from McGill University, where he's a PhD student under the mentorship of Dr. Ahab Abouif. And Juni was selected for one of our Early Career Scientist Awards this semester. And so we're very excited to have him and hear about his research. And so thank you, Juni, for joining us, and I'll hand it off to you.
Thank you so much. And thank you to the selection committee for this amazing opportunity. Can everyone see my screen? Yeah, so today I'll be talking to you about one of my chapters from my PhD on the origin and elaboration of a major evolutionary transition in individuality. So I'm a PhD student in the Abouif lab, and our lab operates under the Eco-Evo-Devo framework. And so we try to understand how development incorporates ecological signals from the environment, such as temperature, nutrition, photoperiod, and how changes in development as a result of the environment, whether it's developmental timing or regulation, could be substrate for selection and the evolution of novel phenotypes. And one of the questions that we try to address is, how do complex biological systems originate and diversify? One of the major paradigms that have been used to understand increases in biological complexity over evolutionary history have been the major evolutionary transitions that were first talked about by John Maynard Smith and E.R. Smashmary, in which case they talk about the instances that have punctuated evolutionary history that have resulted in large-scale increases in biological complexity. That includes the evolution of compartmentalized genomes from independent replicators, multicellular organisms from single cells, eusocial societies from solitary animals, and obligate endosymbiosis from independently replicating organisms. And how this process unfolds is you have solitary units that undergo a group transformation and form a cooperative group. This cooperative group then becomes developmentally integrated through a group transformation that results in the major evolutionary transition to a new higher level unit. And so once a major transition has evolved, you could have subsequent elaboration of the major evolutionary transition. So one example could be the evolution of new cell types within a multicellular system, or the evolution of new workers subcastes in eusocial societies. A hallmark of major evolutionary transition is actually the establishment of a division of labor. And so throughout the genome, you have a division of labor of information across chromosomes. In eukaryotes, which are symbiont unicells, you have the division of labor of metabolism across organelles. In multicellular organisms, you have the division of labor of inheritance through the germline and soma and organ function. And then in social insect societies, you have a division of labor that is reproductive between the queen and her sterile offspring. Now, of the major evolutionary transitions, obligate endosymbiosis represents a dramatic transition in individuality, because you have the coming together of two distantly related taxa, typically bacteria or fungi with an animal, to form an obligate individual. In classic examples, you have coral polyps that have photosynthetic algae that are necessary for their survival and metabolism. And in the case of termites, you have the classic example of trichonympha, which actually allowed them to invade a new ecological niche, which allows them to process wood. And a major question is, how does obligate endosymbiosis originate within a group and diversify within specific lineages? And so today I'll be talking about work that I did with Metin Rafiki, who is a postdoc in the Abouif lab and now has his own group in Istanbul, Turkey, and the obligate endosymbiosis between the bacteria Blockmania and the carpenter ant Camponotus floridanus. Carpenter ants are a hyperdiverse genus of ants. And so worldwide, there are over 1,000 species of carpenter ants, and its mutualism with Blockmania has been described as one of the reasons for their evolutionary success. We Blockmania itself was first described in the 19th century by Friedrich Blockmann, and here you have a drawing from the 1920s by Paul Buchner of Blockmania filling the young oocyte of Camponotus lingipringus, and we could see by comparison in modern confocal micrographs just how striking and detailed these early descriptions were, in this case an oocyte of Camponotus floridanus. The endosymbiosis is believed to have originated around 51 million years ago, where the ancestor of the Campanatini tribe, which encompasses the genera Camponotus, Polyrhachis, and Colobopsis, were infected with the ancestor of Blockmania. When you compare the bacterial phylogeny to the ant phylogeny, we see that they're congruent, which is evidence of a strict vertical transmission over evolutionary time. And in the bacterial phylogeny itself, you have significant gene loss. When we look at a Camponotus colony and we zoom in on the brood, in this case, some first instar larvae, and we see a micrograph, we see something very peculiar in the gut. And so when you look at the inner lining of the midgut, we find that it's lined with hundreds of bacteriocytes, each one of these bacteriocytes containing hundreds of individual Blockmania. And it's known from previous studies that the bacteriocytes produce key amino acids such as tyrosine, which help in their nutrition. We also know from previous studies that loss of Blockmania causes a downward shift in the worker size distribution and decreases colony size. And so when we try to evaluate the social contract that exists between Camponotus and Blockmania, we see that Camponotus provides a protected cellular environment and strict vertical transmission for Blockmania, while Blockmania provides nutritive benefits, it regulates the polymorphism within the species, and it regulates the colony growth and efficiency. And so our first question is, how did the obligate endosymbiosis evolve? And so to understand evolution, we first look at development. And so here you could see the developing oocyte of a Camponotus floridanus worker, in this case, the ovarial. You could see here in the germ stem cells that there's no Blockmania present. And it's only as the oocyte is specified that you have an infection of Blockmania from the somatic ovarial sheath. And once it infects the ovarial, it gets restricted to the oocyte. As the oocyte matures, it gets progressively restricted to the posterior pool. And then at the posterior pool, it surrounds germplasm. Now, germplasm is a specialized cytoplasm that classically from Drosophila and Nozonia was has two functions. One in posterior specification, so its position within the oocyte says that this will be the posterior, which will develop into the abdomen, and it specifies germ cells. And so the plasm that gets nucleated will then develop into the adult ovaries or testes. And so we wanted to first understand whether Blockmania is actually influencing the Camponotus germplasm. And so to do this, we looked at the posterior group genes, which are part of the germline gene network, in this case, Vazin, Nanos, and Oscar. And we first looked at them in Lasius niger, which is a cousin of Camponotus from the same subfamily, the Firmicinae, and it does not have an obligate endosymbiont. And what we find is that all three of these genes localize to the posterior pole, where you would normally find germplasm in Drosophila, and they're localized together. When we look at Camponotus floridanus and we look at a mature oocyte, we see another germline gene, aubergine, is similarly localized to the posterior pole. However, as this oocyte transitions into a freshly laid egg, which is still a single cell, we see a dramatic rearrangement of this gene expression such that it's accumulated in 4 distinct subcellular locations in the embryo. And this is quite dramatic because between this late oocyte and stage 1 embryo, no new maternal products are dumped into the egg as the nurse cells undergo apoptosis. And so this represents a dramatic reshuffling of existing gene products. And unlike Drosophila and Lasius, these germline genes are not expressed together. They're actually expressed in combination. And so here you have the germline gene smog, which is in 4 zones, similar to aubergine, whereas Nanos is only expressed in 2 of the 4 zones. and Oscar is only expressed in one zone, which is the polplasm at the posterior pole. As we go through development and we go from a freshly laid egg to the cellular blastoderm, we also see that, unlike Drosophila and Lasius, that this expression is actually dynamic. Nanos expression changes from 2 zones to 3 zones, and Oscar expression changes from 1 zone to 2 zones. And so we wanted to then understand how the evolution of these 4 localization zones has affected embryogenesis. And so when we look at the standard ant body plan, in this case, again, Lasius niger, you could see 3 major cell types, well, cell lineages. You see the embryo, which is in cyan, that's in the posterior compartment of the embryo. You see the extraembryonic tissues, which are these support cells that will form the amnion and serosa, and these are at the anterior. And you have the germline forming primordial germ cells at the posterior pole in the posterior end of the developing embryo. When we look at Camponotus floridanus, we see that this body plan has been radically altered. And so the embryo itself forms a germ disc at the anterior pole, completely opposite from Blockmania, which now occupies the entire posterior pole. The germline forms in a novel cell type called a germline capsule, which is adjacent to Blockmania. And the germ cells themselves are now spatially uncoupled from the developing embryo and the embryonic posterior. And they're spatially separated by the extraembryonic support structure. And so the egg itself is just dramatically different in how the different cell lineages are set up. And when we get to a later stage in segmentation, we see that the germ cells develop within this germline capsule. And we know that this germline capsule then becomes part of the somatic gonad. And so we wanted to understand, what role do the 4 zones play in actually setting up these radical rearrangements? Is it just that you have 4 regions of germline genes that you end up having 4 germ cell regions, or is there a functional distinction between these 4 different zones? And so to do that, we tracked VASA and protein mRNA localization and expression throughout development by looking at their expression in fixed embryos over a tight developmental time series. And so here in the freshly laid egg, which is stage 1, it only has 2 nuclei, it's a single cell. You see that VASA is expressed in 4 discrete locations. As this freshly laid egg develops into the cellular blastoderm, you can see that each of these 4 localization zones now elaborates. Zone 3 is expressed throughout the entirety of the developing germ disc, which is the developing embryo itself, and it has a concentration at the future site of gastrulation. You see that zone 4 forms a crescent that's abutting the innermost lining of the yolk sac, and that this extends across the contour of the yolk sac into the bacteriocytes themselves. And we believe that this expression pattern is actually important for guiding the bacteriocytes to line the midgut of the developing embryo, where they will eventually be located in the larvae. In zone 2, you could see an increase in the concentration of VASA expression where the novel germline is being formed. And something that's peculiar is really what's happening in zone 1, because even though it's filled with bacteriocytes, the expression pattern of VASA is similar to how germ cells would form in Drosophila or wasp like Nasonia. And so when we look at what's happening in zone 1 by shifting our focus to Oscar, which is only initially expressed in zone 1, we get a better understanding of what's going on within these bacteriocytes. So Oscar forms the germplasm at the posterior pole. During the initial nuclear divisions, this germplasm begins to form multiple foci. And as we enter the cellular blastoderm stage, it actually disperses dorsally, where each one of these foci becomes the center of a bacteriocyte. And this is what we call zone 1B. Zone 1A is an accumulation of Oscar and VASA and other germline genes, which are actually where the germ cells would be located in any other holometabolous insect like Nasonia or Drosophila. And actually during these stages, you could see the recruitment of nuclei into a ridge where the novel germline is actually beginning to form. And this is the functional germline of the host. And we see when we look at higher resolution that each of the bacteriocytes strongly expresses Oscar. And so for this, it's telling us that there's been a co-option of germplasm foci that may regulate bacteriocyte patterning. As we get to gastrulation, we see that now the expression within zone 3 is restricted such that VASA is now expressed in the mesoderm. You continue to see VASA expressed in the lining of the yolk sac into the bacteriocytes. And now within the germline capsule, the novel germline is beginning to develop more, where you form progenitor germ cells that are inside the capsule. And it's at this stage where we see the segregation of a soma and germline bacteria population. You essentially have two populations of Blockmania in the embryo. You have one in the bacteriocytes, which will become the lining of the midgut, and this has strong Oscar expression, and each bacteriocyte contains hundreds of Blockmania strands. And then within the novel germline, you actually have maybe a couple of hundred of bacteria that will be inherited in the next generation. And so the difference in the number of Blockmania between zone 1 and zone 2 is probably more than 100-fold difference in bacteria population. And as we get to germband extension, we find that the ancestral germ cells begin to dissociate and they are eventually undetectable here in VASA and Oscar. And the germ cells that were in the germline capsule now migrate out of it and become incorporated into the last abdominal segment of the extending germband. In this position, they will continue to undergo gonadogenesis. You get VASA expression within the nervous system, which is conserved with other insects. But interestingly, you could see VASA expression in the lining of the midgut where the Blockmania is now localizing. As we get to germband retraction, we get the formation of the gonad, and through dorsal closure, we get its coalescence and migration to its final location. And so from all this information altogether, we can see that we have 4 functionally distinct zones of germline gene expression. We have zone 1A, which is a non-functional ancestral germline. We have zone 1B, which is germplasm foci that may pattern Blockmania in bacteriocytes. We have zone 2, which is the functional novel germline. We have zone 3, which is expressed within the mesoderm and embryonic posterior. And you have zone 4, which is this crescent that we believe is important for guidance of bacteriocytes into the midgut. And now that we have a better understanding of the radical rearrangements, we wanted to understand more deeply what are the developmental genetic changes that are actually underlying the novel zones of embryogenesis. And so to do this, we have to actually go all the way back to the beginning. I mentioned that the obligate endosymbiosis evolved 51 million years ago, and it's predicted that this evolved from an existing ecological mutualism that existed between the ants and hemipterans. Hemipterans harbor many endosymbionts, some of which are exuded through their sap, which the ants feed on. It's believed that there was an ecological horizontal transfer of the secondary symbiont from the sap of the hemipteran bug, which then was isolated within the ant and evolved eventually into the ancestry of Blockmania. We know that in hemipteran bugs, it's actually the Hox genes UBX and ABDA that are actually expressed in and regulate bacteriocytes. So in this case, when you have a knockdown of UBX expression with UBX RNAi, you completely lose the formation of bacteriocytes. And when you knock down ABDA, you get misspecification of the bacteriocytes where they can't coalesce. And so we wanted to know whether ABDA and UBX play any role in the obligate symbiosis in Camponotus. And so again, just like before, we wanted to see how these genes are expressed in a species that does not have an obligate endosymbiont. So in this case, Lasius niger. And we find, as one would expect for insects, that in this case, ABDA begins its expression during segmentation, where it will give the abdominal identity to the segments across the anterior posterior axis. When we looked at Camponotus floridanus, we saw something truly shocking, where these Hox genes, ABD and UBX, are actually maternally localized in the embryo. Not only that, but they're actually localized to the same 4 zones that we found the germline genes. In this case, ABDA is expressed in germplasm in zone 1 and in zone 3, which is similar to how nanos is expressed. And UBX is expressed in all 4 zones, including germplasm, similar to VASA and aubergine. And when we perform a co-localization experiment using VASA antibody and UBDA antibody that recognizes both ABDA and UBX protein, we see that in fact you have co-localization of these 2 genes within each of the 4 zones. And so this is telling us, and so we then wanted to know whether the Hox genes are actually interacting with the germline genes in the 4 zones. And what is the effect on the embryo if we were to knock down these Hox genes? And so it's important to know that across other arthropods, when you normally knock down a Hox gene, you get a shift in segment identity. And so in this case, in a true bug, if you were to knock down UBX, you get the conversion of the 3rd thoracic segment to the 1st thoracic segment into the 2nd thoracic segment. So T3 and A1 would all become T2. And so you get a shift in identity of the limb. If you knock down ABDA, you'd actually shift identity of abdominal segments A through 8 into the 3rd thoracic segment. And so you get multiple legs forming across the abdomen. And so we wanted to see what would happen in the ants. And so we injected syncytial embryos with ABD and UBX RNAi. But unlike what's been observed across any other arthropod, including crustaceans, instead of segment identity shifts, we actually saw severe truncations in the abdomen, where the abdominal segments would be truncated up to the boundary of the expression of these specific genes. And this phenotype closely mirrors the classic posterior group gene phenotypes that were detailed by Ruth Lehman and Noslin Volhard, showing that mutation in germline genes such as Stalfin, Nanos, VASA, and Oscar similarly results in a severe truncation of the abdomen. And with respect to the bacteria, we see that ABDA RNAi results in the complete loss of the bacteriocytes, where no bacteria is detected with, no Blockmania is detected within the embryo. And UBX RNAi results in a mislocalization of the bacteriocytes. And what's intriguing is that this is the opposite pattern of what's seen in true bugs, where normally UBX RNAi results in the complete loss of bacteriocytes and ABDA RNAi results in the misspecification, which is very interesting. We're also able to perform multiple concentrations of our RNAi injection. And we found that when you injected a higher concentration of ABD RNAi, you actually developed a secondary phenotype. So in addition to the truncation of the abdomen, higher concentration of ABDA RNAi results in a bifurcation of the embryonic midline, such that the thoracic segments are forming on either side of the yolk sac. And this can be partially explained by the expression pattern that ABDA and VASA have during gastrulation. And so while ABDA and VASA during cellular blastoderm are initially expressed throughout the embryo, they become restricted to the mesoderm and the ventral midline. We also found that in addition to this, Hydra phenotype where you got the split of the head across either side of the yolk sac, that the high concentration of ABD RNAi also results in severe gastrulation defects, where instead of an extending germ band, you have an undifferentiated stub, which is an ungastrulated germ disc, at a stage which normally should have full germ band extension. And we found that this gastrulation defect actually coincides with the loss of germline gene expression from the germ disc. And so here in a YFP injected embryo, you see germ cellus protein expressed in yellow and in orange, which is a posterior group gene. You can see that it's expressed in a cell population in the lateral ectoderm and in the ectodermal plates. When you have the bifurcation phenotype, you lose the lateral ectoderm expression, but you keep the ectodermal plate expression. But when you get the severe phenotype, which is an ungastrulated embryo, you lose both expressions, and germ cellus just accumulates at the posterior end of the germ disc. And so this tells us that Hox and germline genes not only interact, but have new roles in gastrulation and midline patterning. And so we then wanted to get a more resolved answer about whether Hox genes are actually upstream of the germline genes. And so to do this, we performed a tissue-specific qPCR where we injected embryos with either YFP, ABDA RNAi, or UBX RNAi, and we let them develop to the Germband Extension stage, which is stage 8. At this stage, we could actually dissect out the different tissues of the embryo. So we could dissect out the capsule, which is zone 2. We could dissect out the bacteriocytes, which is zone 1. And we could dissect out the germ disc attached with the yolk sac, which is zone 3 and 4. And we could see how germline genes are expressed across each of the zones in response to knocking down the 2 Hox genes. And we found that across the board from zone 1, 2, 3, and 4, that both ABDA and UBX RNAi resulted in a decreased expression in these transcripts, telling us that ABDA and UBX are actually positively regulating the germline genes. And so this is actually evidence that there's a rewiring of the segmentation hierarchy. And so that genes that normally come on towards the end of embryogenesis to specify segment identity are now maternally inherited, where they control primary body axis specification. And in this case, the formation of the abdomen. And so now that we had a pretty decent idea of the developmental genetics that are underlying this symbiosis, we wanted to address the elephant in the room. With all these developmental innovations that are happening in Camponotus floridanus, what is Blockmania actually doing? Is it a driver or is it a passenger throughout all of these changes? And when one considers that in a freshly laid egg, over 97% of the genomic content is bacteria, and only 2.6% is actually the host, the question becomes how, not if, Blockmania regulate Camponotus embryonic development. And so to test this, we treated whole Camponotus colonies with rifampicin, which is an antibiotic that's known to eliminate Blockmania from bacteriocytes. Here you could see that in the rifampicin treated colonies, the bacteriocytes at the posterior pole are now completely devoid of Blockmania, and we wanted to see the effects on germline and Hox gene patterning. And what we found is that the 4 zones themselves are not affected by the loss of Blockmania. And so here, when we look at vasoprotein, we can see that each of the 4 zones is still specified. And so that this tells us that it's a host process, that the host is actually setting up these zones of Hox and germline gene expression. But as we go from the stage 1 embryo to the stage 6 embryo, we actually see changes in the gene expression. And we actually see 2 different phenotypes that occur in equal probability. So 50% of the phenotypes are mild and 50% of the phenotypes are severe and embryonic lethal. So in the mild phenotype, you have an embryo that structurally and phenotypically looks normal. It looks just like the wild type embryo, except now you have a missing VASA gene expression within zone 2. And so the novel germline no longer forms, and the ancestral germline is present. And in these cases, this ancestral germline can actually give rise to functional gonads in about 33% of the embryos, whereas the other 66% have scattered germ cells and are sterile. When we look at the severe phenotype, we actually find that no embryo is actually forming. What you see in this huge blob is that you have a giant yolk sac here on the left, you have a giant germline capsule here on the right, which we are able to confirm with markers, and you have the bacteriocytes still forming. But the actual expression pattern of VASA is kind of all over the place. And when you look at the mild phenotype more closely, it actually becomes clear that what Blockmania is doing is that it's selectively regulating specific Hox and germline mRNA and protein within these embryos. And so when we look in the case of ABDA, in the mild phenotype, ABDA is lost from the germplasm at the posterior pole. tutor is lost from the bacteriocytes, and as I just showed, vasa is lost from the novel germline. And so this is telling us that Blockmania is kind of selectively regulating the combination of the genes that are expressed within each zone. And so to go deeper into understanding this, we performed transplantation experiments where we actually took Blockmania from donor wild type embryos, where we picked up cytoplasm from the posterior pole where Blockmanias were located, and we injected them into rifampicin embryos that are from rifampicin treated colonies. And what we found is that the transplantation of Blockmania could actually restore the lost gene expression that occurs when Blockmania is missing. And none of these transplanted embryos developed into severe phenotypes, even when otherwise 50% would have. And so what this shows us is that Blockmania is indeed maintaining and selectively regulating the zones of germline and Hox gene expression, potentially by regulating the combination of genes within each zone that actually makes them functionally distinct from one another as modules. And so the last question we wanted to address is really throughout the evolution of this obligate endosymbiosis. Did all of the developmental innovations that we observe in C. floridanus evolve at the origin of the major evolutionary transition to obligate endosymbiosis, or was there actual gradual developmental evolution happening within preceding lineages prior to the major evolutionary transition? And so to test this, we did an ancestral state reconstruction of 5 developmental characters across 31 species of ant, including 3 different subfamilies, the Formicinae, the Myrmicinae, and the Dulcodurnae. And we reconstructed the character states of the number of germline localization zones, whether or not there was maternal Hox expression, and the number of maternal Hox localization zones, whether the lineage had an independent obligate endosymbiont, the position of the embryo, if it was in the anterior or the posterior, and whether or not the embryo had a germline capsule. And so when we look at the most recent common ancestor of the Dulcodurnae, Myrmicinae, and Formicinae, we find that the most recent common ancestor had 1 germline zone and no maternal Hox expression. And so the germ disc is developing in the posterior portion of the egg. The germ cells are marking the posterior pole and are at the posterior of the developing germ disc, and there's no localized Hox expression. And so this would be similar to what would be seen in Nozonia wasps or Drosophila melanogaster. When we then make our way into the Formicinae, which is the subfamily that contains Camponotus and the Campanotini, we see something entirely different. We actually find multiple pre-existing developmental capacities throughout the Formicinae. And so in this case, in Brachymyrmex patagonicus, we see that the germ disc is developing in the anterior pole, despite having no discernible endosymbiont in the posterior pole. We find that the germ cells are actually developing in the posterior, but it's actually spatially separated from the embryonic posterior. And so what this is telling us is that that classic dual function that germplasm has of both specifying the embryonic posterior and germ cells could already be decoupled in these early Formicine lineages. And this is also in the context of no maternal Hox expression. When we then look at species like Formica occulta, we find the first instance of a novel germline localization zone. In this case, vasa is expressed in the homologue of zone 3, which is the embryonic mesoderm, and it's also in the germ cells. And this is also the first instance of maternal Hox expression. And what's truly fascinating here is that maternal Hox genes are first localized in a somatic area of the embryo, which is zone 3. Zone 3 is part of the embryonic patterning of the mesoderm, ventral midline, and embryonic posterior, but has nothing to do with the germ cells. And so the first instance where these 2 gene regulatory networks interact are within zone 3. And this may have actually facilitated the inheritance of Hox genes within the germ cells at the origin of the major transition in the Campanotini. And so when we look closer at these Formica lineages, we actually see that in Formica and another species of ant that we looked at, Anopolepis gracilipes, that these lineages actually have independent evolution of an obligate endosymbiont. They have a symbiont that is phylogenetically distinct from Blockmania, so they're independent infections. But in both cases, you have maternal Hox gene expression and an extra germline localization zone. And when we look at the endosymbiont, we see that similar to Blockmania, it surrounds germplasm at the posterior pole. And this suggests that Hox and germline and novel germline zones may be a general feature of ants with obligate endosymbionts and may be necessary for the evolution of the developmental integration of symbionts within ant embryos. And so now we get to the Campanutini, which is the tribe that's infected with Blockmania. And when we look at two genera, Polyrhacus and Colobopsis, which are more basally branching than Camponotus, we see that instead of 4 zones, you now have 3 zones of germline and maternal Hox gene expression. You get the evolution of zone 4, which again, we believe is important for bacteriocyte guidance into the midgut. And you have zone 1, which is expressed in the middle of the bacteriocytes, and we predict is involved in patterning the bacteriocytes and acting as a germline all at once. And it's in these lineages that we see for the first time the expression of Hox genes within germplasm. And so these are innovations that are definitely at the origin of the obligate endosymbiosis, the germplasm expression of Hox genes and the evolution of zone 4. And what this tells us is that the evolution of zone 2, which is the novel germline, actually evolved to increase the efficiency of symbiosis by dividing labor. And so in zone 1, you have germ cells that have to form germ cells and pattern bacteriocytes in Polyrhacus and Camponotus. Whereas here in Camponotus floridanus, you have zone 2, which is now a dedicated germline. And so you could have this non-functional germline in zone 1 left to the devices of Blockmania, and you could form your germ cells independently. And it's at this point in the phylogenetic tree that we see expression of Hox genes within zone 2 as well. And so what this tells us is that the origin of obligate endosymbiosis between Campanutini and Blockmania required the co-option of pre-existing developmental capacities that evolved not for the purpose of obligate endosymbiosis per se, but were necessary at the origin in order for developmental integration of Blockmania into Campanutini. And as for our question, it's telling us that there was a stepwise addition of zones from 1 to 2 to 3 to 4, which would support a gradualistic developmental evolution across the advanced ants, as opposed to this saltation in developmental characters at the origin of the major evolutionary transition. And so within the broader themes of my thesis, it really makes me think about the ability of tinkering with the germline for evolutionary innovation in ants. And so in this case, we saw how the evolution of novel localization zones of germline gene expression into 4 modules within the embryo allowed this developmental integration of the obligate endosymbiont. And within ants in general, I'm starting to see that there's a remarkable variation in germline development. So if we look at 2 distant lineages of ants, Odontomachus, which is part of the Poneroid clade, and Novomeser, which is in the Myrmecines, which is in advanced ant society, we see huge variation in germplasm morphology, where the Odontomachus germplasm takes over 30% of the embryo. Through development, we see that this variation in germplasm morphology results in variation in germ cell formation. And so now these germ cells are actually a second compartment of the embryo where normally the extra embryonic material would be, but instead they're all germ cells. And when we see the actual gonad size in another Ponerine here, Harpagnathos, we see that these germ cell formation differences allow these Ponerines to have 45 to 100 germ cells per gonad, whereas other ants like Brachymyrmex, Patagonicus only have 5 to 10 germ cells. And then there's the curious case of obligate sterility. And so obligate sterility is a character that's evolved multiple times across ants in both formicoid and poneroid clades. And this involves having workers that have no ovaries at all. And so normally workers have ovaries that are repressed by the queen through her pheromones. But in lineages that are obligately sterile, they have no embryos, and during embryogenesis, you actually can't detect germ cells. And what we found in these species, in this case, Monomorium furonis, is that you actually have two developmental programs of germ cell migration. Here on the right, here on the left, you have the queen. And as the video loops, you can see the arrow is pointing at this cluster, which is internalized, which is the germ cells. In exactly stage-matched embryos, this cluster is excluded from the embryo in worker-destined eggs. And so workers have a germ cell cluster that is outside of the egg, and queens have a germ cell cluster that is inside the egg. And so you have this in or out germ cell cluster. And the result of this is you have an embryo that could form a gonad and an embryo that is completely sterile. And so you have this embryonic caste determination through germ cell loss. And so variation, which we see in germline development across ants, really is the substrate of selection. Where here in the case of obligate endosymbiosis or reproductive division of labor, you have this adaptive evolution through germline modification. And so with that, I'd like to thank my supervisor, Ehab Abuhif, my collaborator, Mateen Rafiki, who is instrumental for all this work, and the rest of the Abuhif lab. And I'll be happy to take any questions.
Thank you, Junie. That was wonderful. We do have some time for questions. So feel free to turn on your mic or put them in the chat.